The first step in DNA replication is unzipping the double helix structure of DNA by the enzyme helicase. Hydrogen bonds are broken by helicase, which hold all of the complementary bases of DNA together. The separation of these two strands creates the fork, and this replication fork allows for RNA to copy one side of the strand. One of the strands is oriented in a 3 to 5 prime direction towards the replication fork. This is the leading strand. The other strand is oriented to the 5 to 3 prime direction, which is the lagging strand. As a result, these two strands are replicated differently due to their different directions. The leading strand has a short piece of RNA called RNA primer. This is produced by primase, and it binds to the end of the leading strand. The primer acts as the starting point for DNA synthesis. DNA polymerase binds to the leading strand and walks along it, adding new complementary nucleotide bases to the strand of DNA in the 5 to 3 prime direction. The lagging strand has numerous RNA primers, and they're made up of the primase enzyme and bind at various points along the lagging strand. Chunks of DNA called Okazaki fragments are then added to the lagging strand in the 5 to 3 prime direction. This type of replication is called discontinuous, as the Okazaki fragments will need to be joined up later. Once all the bases are matched up, A to C, C to G, an enzyme called exonuclease strips away all the primers, the gaps where the primers were, and then filled by yet more complementary nucleotides. This U strand is proofread to make sure there are no mistakes in the DNA sequence, and DNA ligase then seals up the DNA into one continuous double helix. The result of DNA replication is 2 DNA molecules, one consisting of the new replicated DNA and the old chain of nucleotides that was replicated from. Before transcription can even take place, DNA must unwind at a specific area called the transcription bubble. This transcription bubble is the site in which the specific gene which is wanting to be replicated is targeted. Transcription uses one major enzyme, which is RNA polymerase, which handles the majority of the transcription process. The transcription uses 2 exposed DNA strands due to its unwinding of the double helix. One is called the template strand, which is directly complementary to the RNA polymerase, and the template strand is completely identical to the non-coding strand, which is the opposite strand to it. The only difference between these two strands is that the RNA polymerase will be adding new U nucleotides instead of T's. The site of DNA from which first RNA nucleotides is transcribed onto is called the +1 site, or the initiation site. Nucleotides that come before the initiation sites are given negative numbers and are said to be upstream. Nucleotides that come after the initiation site are marked with positive numbers and said to be downstream. RNA polymerase is the specific enzyme that transcribes DNA to RNA. RNA polymerase binds to the DNA gene region called the promoter. The promoter tells the polymerase where to stay and begin transcribing on the DNA. Once the transcription bubble has formed, the polymerase immediately starts transcribing. In eukaryotic organisms, the main promoter area which the RNA polymerase binds to is the TATA box. The RNA polymerase cannot directly attach itself to the TATA box, so it must use helper proteins called basal transcription factors, which bind to the promoter first, helping the RNA polymerase attach to the cells. Elongation is where the RNA polymerase walks along each strand of DNA as a template strand in the 3 to 5 prime direction for each nucleotide in the template. RNA polymerase adds a matching complementary RNA nucleotide to the 3 prime end of the RNA strand. The RNA transcript is nearly identical to the non-template coding strand of DNA. RNA polymerase will keep transcribing until signals tell it to stop. The process of ending transcription is called termination, and it happens after the RNA polymerase transcribes a sequence of DNA known as a terminator. Translation initiation is made up of 3 parts: the ribosome, the mRNA, which was coded in transcription, and the initiator tRNA carrying MET, which is a start codon for the protein. With these three components, they meet together in just the perfectly right way and create the initiation complex, which begins building the first protein. The initiation complex begins with the tRNA carrying MET attaches to a small ribosomal subunit. They bind to the 5' end of the mRNA, recognizing the 5' GTP cap. Then they walk along the mRNA in the 3' direction until they reach the start codon, which is always AUG. Once the MET codon is placed onto the mRNA strand, this allows for elongation to begin, which is the elongating of the polypeptide chain. This begins with the first tRNA starting off in the middle slot of the ribosome, in the P slot in the EPA, and then 3 slot called EPA. Next to it, a fresh codon is exposed in another slot called the A site. The A site is the landing site for the next tRNA. One anticodon is the perfect complementary match to each exposed codon for the tRNA. Once the tRNA has landed in the A site, the formation of the polypeptide bonds that connects one amino acid to another begins. And the transfer of MET from the first tRNA of the amino acid then is transferred to the second tRNA in the A site. Once the polypeptide bond is formed, the mRNA is pulled onward by the ribosome by exactly one codon. This shift allows for the first empty tRNA to drift out of the E or the exit site and exposes a new codon in the A site where the whole cycle can repeat. It can repeat over and over, and it can take over thousands of repetitions in order to create a polypeptide chain, which will create a protein. The eventual end of this entire process is termination. These polypeptide chains must come to an end, and translation ends in this process called termination. Termination happens when a stop codon in the mRNA, which consists of UAA, UAG, and UGA, enters the A site. These stop codons are then recognized by proteins called release factors, which fit neatly into the P site. They aren't tRNAs, however, the release factors mess with the enzyme that normally forms polypeptide bonds. These make it add a water molecule to the last amino acid in the chain. This reaction separates the chain from the tRNA, and the nuclear protein is then released. After all this, the small ribosomal subunits that were used to separate the mRNA from each other, each can take part in another round of translation in order for this process to happen over and over for the creation of more and more proteins.